Today we are going to see the structure of proteins. We have already seen the structure of amino acid. So first we are going to see the today structure of protein. Structure of proteins. Before going to see the structure of protein, students, you should know the structure of peptide bond. First we will see the structure of peptide bond. Peptide bond. First we will see the structure of peptide bond, then only we comes to know the structure of proteins. Now what is peptide bond, students? Peptide bond, or we can say peptide linkage, is an amide formed between the group, this is the carboxylic acid group, and amine group NH2 by elimination of water molecule. This is about it is the definition of peptide bond. Now students, first we will draw two amino acid. First amino acid, students, this is the carbon, which is tetrahedral structure. It has got a acidic group and it has got amine group, and this is alkyl group. It may be same or different. Now second amino acid I have taken, students, NH2 I am writing like this. This is the carbon which has tetrahedral structure, acidic group, and this is what? Alkyl group. Now what happens, students? This OH of the carboxylic acid react with the hydrogen of the amines, and it will gives you water molecule. It will eliminate water molecules by heating. So these two are amino acid. This is also amino acid. On heating they will eliminate water molecules and gives you, it will form the amide formation. How it will form, students? Here CO can be written as CO, NH I can write like this, NH, and this is carbon which is tetrahedral structure. This side acidic group, and this is alkyl group. Same thing is, students, this side you have carbon which is tetrahedral structure, alkyl group. Here it is what? Amine group. So it is forming the amide formation. This is the amide formation. This bond is because of the amide formation. Amide has been formed. This is called as peptide bond, because the two amino acid, they will form the peptide bond. This we call as peptide bond. When it will form the amide, it will call as what? Peptide bond. In peptide bond, there is a formation of amide formation is there. Now students, when it will form the amide chain, students, again it will remove the water molecule from this side and one hydrogen from this side. So this is from the amide chain, peptide chain. This way it will form the peptide chain. This peptide chain, when so many chain comes together, it will form the proteins molecules. Now students, depending upon the number of amino acid, depending on the number of amino acid molecules composing a chain, the peptides may be so many types. They may be dipeptide, In dipeptide, students, it contains two amino acids unit. Sometimes it will forming the tripeptide. At that time it contains three amino acid unit, and so on, and it will form last polypeptide chain contain more than 100 amino acid. Proteins are, example students, insulin. When you see the example, insulin is a protein which contains 51 amino acid. So therefore students, we can say proteins are basically polypeptides. Now students, we are going to see the structure of proteins. Now we can explain the structure of proteins very well. The first structure, students, of protein is, first one is primary structure. Primary structure, students, which chain we call as primary structure? When the sequence of amino acids held together by peptides bond, so many peptides bonds. In linear now students, in primary structure we have thread-like structure. This is the primary structure. It has only length is there. This is called primary structure. If you draw the chemical form, it will be like the chemical bond will be like this. This is the peptide bond, students. The first amino acid, the second amino acid I am drawing like here. Second alkyl group here. The different alkyl groups are there. Therefore, different proteins will form. So this is the structure, students. Because of that, now students, we got because it had got only one dimensions, so that is why it is called as primary structure. It is called only linear structure. Because of this linear structure, it is called a primary structure. Here the side chain R1, R2 and R3 will make different proteins. Molecules. It has got only one dimensions only length is only the dimension. That is why it is called as primary structure, because it has only length. That is why it is called as primary structure. Now students, we will see the secondary secondary structure, secondary structure, secondary structure, students. Here the hydrogen bonding in linear primary structure when held by the hydrogen bonding. When primary structure, when primary structure of proteins are held by hydrogen bonding, between carbonyl and this amine group occurs resulting in folding of polypeptides. So it will form the secondary structure. When primary structure is held by the hydrogen bonding, it will form the secondary structure. Now students, it has got length as well as breadth. It may be alpha helix or beta plated. The first one we will see the structure. It may be like this. It may be like this. There is two types of helix, students. This is alpha helix. Now here the hydrogen bonding, students. Suppose here carbonyl group will be there, students. It will attach by hydrogen bonding. This carbonyl group is attached by the amine group of this one by hydrogen bonding. Now students, it has got length as well as breadth. This is the length and this side is forming breadth. So it has got two dimension, length and breadth. That is why it is called as secondary structure. Secondary structure is two types. Secondary structure of proteins two types, alpha helix and beta pleated sheets. How sheet is having only two dimensions, same thing is happening in the secondary structure. Now students, tertiary structure we will see. Tertiary structure of protein. In tertiary structure, students, we will find three dimension, length, breadth and height. Tertiary structure of proteins. Alpha helix, when secondary structure, that is alpha helix, bond twist and fold, leading to three dimensions. Three-dimensional folding of polypeptide chain. When secondary structure students with the help of and twist together and forming three dimension length, breadth and height, we call as, here we will find length, breadth and height in the structure. This is called a tertiary structure. Last one, students, it has quaternary structure. When tertiary structure structure of proteins are involved, here to link the various polypeptides chain and complex proteins are associated. So this is called a quaternary structure of the proteins. Now students, now we are going to see the classification of proteins. Classification of proteins on the basis of molecular shape. Now we are going to classify the proteins on the basis of molecular shape. Proteins are on the basis of proteins are classified into two categories. First one, students, first category is called as fibrous proteins. Fibrous proteins. And second we have, students, globular proteins. Fibrous proteins, students, we are having the fibers. We are getting the fibers. If for example, tendons, keratin, hair, skin, nails, horns, feathers. These are coming in fibrous proteins. We are having examples, students. We can write tendons, keratin, which is present in hair. This is also made up of keratin. Skin. So many examples are there. Feather. Now what about in globular proteins? We have, students, we have the structure of proteins present in hemoglobin, albumin, insulin. These are the examples of the globular proteins. So the proteins are classified on the basis of shape, fibrous protein and globular proteins. Now students, we are going to see the denaturation of proteins. What is the denaturation of proteins, students? Denaturation of proteins. Denaturation of proteins. Denaturation means, students, what? Denaturation means uncoil the proteins. Denaturation means uncoils the proteins. Uncoil the proteins and destroys the shape. It's called as denaturation. It is irreversible process. It is an irreversible change. It is brought by heating with alcohol, concentrated acids and salts of heavy metals. Example, students. Example, we have boiling of eggs, conversion. First example, students. Second, we can write so many examples are there in day-to-day life. Boiling of eggs, conversion of milk to curd. This is because of denaturation of proteins. Thank you, students, very much. If you like my video, please share it.